It's really my honor and a great pleasure to introduce today's speaker, Dr. Hollis Klein. So, Dr. Klein is the chairman of the Department of Molecular and Cellular Neuroscience, the Hahn Professor of Neuroscience at the Scripps Research Institute. She received her Ph.D. in neurobiology from the University of California, Berkeley, then did her postdoctoral training at Stanford and Yale. She has received numerous awards, including the NIH Director's Pioneer Award. In 2012, Dr. Klein was named as a Fellow of the American Association for the Advancement of Science. In 2015, Dr. Klein was elected as the President of the Society for Neuroscience. Dr. Klein's research focuses on the mechanism by which sensory experience affects the development of brain structures and function. The Klein lab has repeatedly been at the forefront of innovative technology advances, including in vivo time-lapse imaging, in vivo electroporation, viral express gene transfer, combined with in vivo time-lapse imaging, application of RNA interference to Xenopus. Her studies had led to increased understanding of mechanisms controlling neurogenesis, synapse formation and plasticity, structural development of neurons, and assembly of functional circuits. Dr. Klein's studies have relevance to a variety of developmental neurological disorders, including Fragile X syndrome, Rett syndrome, autism, and schizophrenia. So now I'm going to tell you something about Dr. Klein that you cannot find in the PubMed. As a former graduate student in the Klein lab, I can tell you that Holly is really an exceptional mentor. So because she not only patiently nurtures the scientific growth of her trainees, she also tried to make an environment that made her trainees can enjoy the fun part of the tedious research. So just to give you an example. So my most memorable moment in the Klein lab is when she told me that you cannot graduate if you don't know how to open champagne bottle. I don't know if you still remember, but sorry, Holly, I'm still not very good at doing this now. So now, without further ado, I would like to invite Dr. Klein to the podium, and the title of her talk today is Building Circuits for Visual Information Processing.
Good afternoon. Thanks, Lee, for the kind introduction. Yeah, when Lee's first paper was accepted, we celebrate this by having champagne. And she was fumbling, and it was really hard for her to open. It was clear she hadn't opened a champagne bottle before in her life. So this was actually, an important step forward for her in her training process. So thank you all for coming this afternoon. I'm going to talk about information processing in the visual system and how to build circuits to process that information. I want you to know at the onset this is geared towards a pretty broad audience. I'm going to give quite a lengthy introduction and cover a lot of the history that I think has been very important in this field before I actually get to tell you a few of the stories of the recent research in my lab. So most of our research is done on this animal, Xenopus laevis, as a tadpole. And you can see here that the animal is transparent at early stages of development. And this is key to allowing us to do one of the major methods that we use in our lab, which is in vivo time-lapse imaging. Okay. So as I said, we're interested in how visual information is processed by the central nervous system. And people have been interested in this problem for quite a number of years and centuries, in fact. And I really like this drawing from Da Vinci because it showed that he had some of the basics right. He knew that visual information came in from the eyes, and it entered the brain via the optic nerve. Oopsie. But after that, things were a little bit confusing. He didn't really know where the optic nerves sent their information and how that information was processed thereafter. Over the next couple centuries, we learned a fair amount. I'm going to use this instead. And this is a bioinformatics study done by Van Essen and his colleagues, where basically what they did was they took all the data that was available in the published literature and generated this connectivity map along a bioengineering kind of motif. And what you see here is the retinal ganglion cells send axons through the LGN and into the visual cortex. And then what they realized after that, and what is communicated in this image, is that there is a huge amount of connectivity from visual centers of the brain to virtually every other portion of the brain. And so this was, in my opinion, some of the early connectivity studies that was done. And now there's a lot more connectomics, which have been studied both in the visual system and in other parts of the brain. This shows two different levels of analysis of connectivity in the brain. One is at this macro level, where basically white matter is mapped out in order to get a rough approximation of what parts of the brain connect to other parts of the brain. And by the opposite scale, this is a rendition of a serial EM reconstruction of a teeny-weeny part of one portion of the brain in order to examine the synaptic interactions that occur in one small region of the brain. And the idea is that at some point, we'll be able to go between these two different dimensions and understand the full connectivity of the brain. But there's another old-style kind of connectomics, which is represented in this classical image from the work of Hubel and Wiesel and Simon LeVay. And basically, in this experiment, as many of you already know, what Hubel and Wiesel were using many electrophysiological methods to record from the visual cortex of primates and cats in order to understand how the organization of visual information is processed in visual cortex. And their neighbor down the hall at Harvard at the time, Simon LeVay, was using anatomical methods to look at connectivity. And here, what they did in this collaborative study was inject one eye of the macaque monkey, an adult, with radioactive fucose and amino acids. And what they discovered is that these axons from that visual pathway project into what we now call ocular dominance columns. And the reason why this was important was this was actually one of the first indications using anatomical techniques of a higher level of structure of information within the brain. And this allowed Hubel and Wiesel to interpret many of their electrophysiological studies, which had demonstrated that there were some regions of the cortex which were predominated by input from one eye or the other eye or both eyes. Now, at the time, Carla Schatz was a graduate student in the Hubel and Wiesel lab, and she went down the hall to talk to Simon, and he said to her, well, you know, those ocular dominance stripes, they're not present in young animals, in newborns. And so this is an experiment which demonstrates that in kittens, there are no ocular dominance stripes seen when one eye is injected with these radioactive compounds. And gradually, over the first few weeks of life, the animal acquires these ocular dominance columns as shown here. So what is it that results in the acquisition of this type of organization within brain connectivity, within the visual system? So again, Hubel and Wiesel were interested in addressing this question. That they used initially macaques, where they sutured one eye closed and then tested the consequences of blocking input in one eye on the output of these visual connections. And what you see here is, again, a classic result from these studies where basically what they discovered is by injecting the radioactive compounds into the open eye, they found that those connections took over a large part of cortical space, as you see here by this predominance of the white label, the autoradiographic label in visual cortex. So again, this was a fundamental observation, a Nobel Prize-winning observation, in fact, that led to many different experiments exploring what mechanisms might operate in order to establish these rules and patterns of connectivity within the central nervous system. So to jump ahead, a framework which has been extremely valuable for many of us in order to understand this type of plasticity in terms of the development of connections within the brain and how sensory input, like monocular deprivation, can result in changes in connectivity over time. One of the frameworks that's been extremely valuable was presented by Donald Hebb, and then the modern reincarnation or restatement of the Hebbian plasticity model in spike timing dependent plasticity, or STDP, shown here. So in 1949, Donald Hebb, thinking about basically what happens in our brains during sleep, came up with this idea, which is now sort of the basic idea about Hebbian plasticity, which is considering, he was considering when one cell was connected to a single postsynaptic cell. So, and what he postulated was that when that presynaptic cell is close to a postsynaptic cell and takes part in repeatedly causing that postsynaptic cell to fire, something changes, some metabolic or growth event occurs so that the connections between cell A and cell B, the efficiency of cell A in driving cell B is enhanced. Okay, so that was in 1949, and that was good. And then a couple decades later, Gunther Stent and von der Marlsberg, more or less independently, thinking about how this Hebbian rule might operate within sensory systems, and specifically within the visual system, they added two important elements to this Hebbian idea. And one was, now, instead of just considering a single presynaptic cell, we're going to talk about convergent inputs terminating on a same postsynaptic cell. And what they offered, the way that they modified this Hebbian idea was to say that when those two convergent inputs are co-active and participate in repeatedly firing the postsynaptic cell, then those inputs would be strengthened. And if those inputs are not correlated in their activity in driving the postsynaptic cell, then those synaptic connections would be either depressed or lost. So those were two important modifications. One is the strengthening and depression of the synaptic contacts, and the other is this idea of convergent coactive input. And again, that was good. But a few decades later, Henry Markham and Larry Abbott, and in fact many other people, identified what we now call spike timing dependent plasticity rules. And this also added more depth and flexibility to the Hebbian rule by saying that that it was the relative timing of activity between the pre and the postsynaptic cells which would influence whether or not the synaptic strength was increased or decreased. And I'd just like to point out that there's this great review about the different cellular mechanisms that can carry out these spike timing dependent plasticity rules by Dan Feldman in Neuron several years ago. All right, So again, that was a very valuable framework for how to think about plasticity within the central nervous system. And now this framework has been applicable to many different experimental systems. And so now I'd like to talk about some experiments that have been done in the frog system and go back and forth between what we've learned from the frog and corroboration with other experimental systems. So again, the frog visual system is a little bit simpler than in cats and primates. So the retina projects to the contralateral optic tectum, and the tectum, which is analogous to your superior colliculus, is basically where all the visual information processing occurs in these animals. One interesting aspect of these animals is that they are growing as we get to look at their visual system. And initially during development, the topographic map, which is a representation of space within the optic tectum, is not very well organized because the optic tectum is pretty small and the axons are all squished on top of one another. So there is no topography at these early stages of development. But as the animal grows larger, the optic tectum gets bigger and the axons can spread over the space. So now there's an opportunity for topographic map formation to be established. And so many of the experiments I'll tell you about are about the development of this topography. So as Lee mentioned, I did my postdoc in Martha Constantine Patton's lab, and when she was a graduate student, she did this, she was interested in axon guidance and how it is that the optic nerves found their target. And in so doing, she took eye primordia from frog larvae and transplanted them or implanted them along the rostrocaudal axis of the embryo. And what she discovered, quite by accident, was that if she implants the eye in the head region of the larva, then it will extend an axon, an optic nerve, into the normal target area, the optic tectum, and share that available target space with the normal input from the other eye. And what happens, what she noticed, was that the two eye inputs don't overlap, but they segregate out into stripes. And this was highly reminiscent of the ocular dominance stripes that Hebbian and Weasel had recently published. So she took this opportunity as a way to study some of the cellular mechanisms that might be operating during the establishment and maintenance of these eye-specific stripes, in collaboration with her postdoc at the time, Tom Ray. And what they tested was whether or not activity in the eye was required for the organization of their axons way off in the target area. And the way that they did this was by blocking action potential activity with TTX introduced into the eye. And what you see here is the normal stripe pattern in a sham-operated animal, and the loss of that ocular or eye-specific stripe pattern in animals that are treated with TTX. Now, this may seem like a natural conclusion now, but at the time, many people thought that these ocular dominance stripes were actually formed as a result of axon guidance mechanisms that had nothing to do with the activity in the eye. And so this was a very important experiment in terms of pointing people towards what were the salient cues that were involved in the organization of these axonal projections. Tom Ray did another very important experiment, which is highlighted here, and that is that he reconstructed the axon arbor of individual retinal ganglion cells that terminated in the optic tectum of these so-called normal three-eyed animals not treated with TTX, or the ones that were treated with TTX. And what you see here is that as a result of blocking action potential activity back in the cell body, the axon arbors of these neurons were largely expanded. And I think this was probably the first experiment that demonstrated that by modifying the electrical activity in neurons, the structure of that neuron can change. And now, again, this is one of the core principles in how we think about neuronal plasticity. So that was an experiment that was done in frogs, and then several years later, a few years later, Mike Stryker and Bill Harris repeated the same experiment and demonstrated that this same observation could be generated in cats. So here again are the ocular dominant stripes in cats, and when an animal is treated with tetrodotoxin in the eye, it fails to develop ocular dominance columns in visual cortex. And this was great because it strengthened the confidence in this idea that activity from the retina into the central nervous system was a core aspect of organizing those central projections. And then again, a few years later, Antonella Antonini in Mike Stryker's lab did a comparable experiment to look at the axon arbor structure. What she did here is label individual geniculocortical axons in animals in the closed eye pathway or in the open eye pathway. And what this experiment demonstrated was that, analogous to the TTX experiment that Tom Ray had done, she showed that different amounts of visual activity, blocking visual activity or enhancing visual activity, would cause structural differences in the axon arbors within that visual pathway. So what this meant was that when you look at this macroscopic image that Hubel and Wiesel and Simon LeVay had produced in terms of the overall organization of the ocular dominant stripes in the macaque brain, that those structural differences were likely carried out by structural differences at the level of individual neuronal axons and their projections. So one cartoon that can summarize this, again going back to the frog system, is that if in the retina, if we look at, sorry, I just have to find my arrow every once in a while. If in the retina, if we look at two retinal ganglion cells that are neighbors to one another, they see the same part of the visual field. When they terminate in the optic tectum, they're coactive. They would drive postsynaptic neurons similarly, and the synaptic connections between those two cells and their postsynaptic partners would be strengthened. But the blue retinal ganglion cell is looking at a different part of the world. It grows into this area. Its activity is completely different, not coactive with the other inputs in that region. It grows into another part of the target area and terminates there. Now, similarly, if we consider the duly innervated optic tectum, like in the Tom Ray and Martha Constantine Patton experiments, the same rules would apply, where retinal ganglion cells from the two eyes are looking at a different part of the world, and when they grow into the same tectal lobe, they segregate from one another because their activity is different. So this, along with the Hebbian hypothesis, we interpreted this to mean that coactive synapses are stabilized, non-coactive synapses may be made, but they're rapidly retracted because they're not strengthened and stabilized. And then another part of the piece in this puzzle was the identification of the postsynaptic, the component in the postsynaptic cell that detected the convergent activity and resulted in the stabilization of these afferent inputs. And that was demonstrated to be the property of the NMDA receptor on optic tectal neurons. And by blocking NMDA receptor activity within the optic tectum, eye-specific stripes were desegregated. Now, this experiment was also quite interesting because the way that I did this experiment when I was in Martha's lab was by implanting a piece of plastic over the optic tectum that would slowly release this drug, APV, which would block the NMDA receptors. And in animals that were siblings to the one that you see here, I could go in and remove that plastic so that the animals were then reared for a further period of time in the absence of drug. And what happened in that case was that those animals then re-established eye-specific stripes. So for me, that was a very important observation because it meant it highlighted the fact that these axon arbors are capable of moving around all the time, and they could be forced to segregate and desegregate depending on the activity of the NMDA receptor on the postsynaptic cells. So after that, I said, I'm not working with any more fixed tissue. So these experiments were all, you do the manipulation, you fix the animal, you do a histological assay and see if there are stripes or not. Thereafter, I wanted to completely do experiments using in vivo time-lapse imaging so I could actually look at the dynamic rearrangements of these axons as they're occurring in response to visual activity. So in this movie, what you see are several retinal ganglion cell axons, which are shown here labeled in red, and their presynaptic sites, which are labeled with synaptophyes and CFP. And this is a movie that was where images were collected at 10-minute intervals over the period of about an hour. And what you see here is that these axon arbors are moving constantly over a period of about an hour. And so we were interested in how quickly synapses could form and disassemble in the intact animal. And the way that we did this was, again, by using those same reagents, the cytosolic fluorescent protein and the synaptophyes and CFP, and we collected images over time. And what you see here, so this is the YFP, and this is the synaptophyes and CFP. And now this is a chronogram, and to see the results most clearly, let's start at the end of the time series. And what you see shown by these arrows are two presynaptic puncta. And if we go backwards in time, you can see at the beginning of the observation period, those synapses, that presynaptic machinery was not present. So what this told us is that over the period of about an hour, synapses can assemble before your eyes in the intact animal. But the other thing this said is that the blue punctum that you saw there at that arrow at 70 minutes was not as bright as these other puncta, indicating that synapses can begin to form within an hour, but they don't develop their full strength. So Ed Ruthazer, who was doing these experiments, then imaged over a longer period and found that basically synapses would acquire their full maturation within a period of about 6 or 8 hours. We also had evidence that it was very clear that branch retraction was actively involved in the elaboration and dynamic rearrangements of these axon arbors. So basically, what this suggested to us was that as the axon arbors are innervating the optic tectum, they're extremely dynamic. They send out exploratory branches, and these branches can form synapses that are coactive with other convergent inputs. And in the rare cases where those dynamic branches are coactive with their neighbors, activate NMDA receptors, then those synapses will be stabilized and they'll serve as the basis by which the arbor is further elaborated. So all of that fit in very well with this, again, this framework of spike timing dependent plasticity as being a core rule for the regulation of synapse formation and stabilization, and how synapse formation and stabilization would participate in the elaboration of axon arbors and the development of topographic maps. But over the years, it became clear that there was a dark side to spike timing dependent plasticity, and many labs had reported that in cases as shown here, when a receptive field of a neuron was repeatedly stimulated with a single directional stimulus, that receptive field would shift towards the origin of the direction of origin of that stimulus. And so what that indicated was spike timing dependent plasticity rules can't be the whole story in terms of topographic map formation, because the topographic map would shift in the direction of predominant visual stimulus. And so Masaki Hiramoto, who's a postdoc in my lab, did a series of experiments to try to explore if there might be other mechanisms that operate along with spike timing dependent plasticity that would facilitate topographic map formation. And what I'm going to tell you is about the evidence that he has for a temporal code of activity in the retina, which is communicated to the tectum, and that this code instructs map plasticity. So Masaki wanted to find out whether or not there's information in the natural scene that would instruct the development and maintenance of topographic maps. And to do that, he stared at his animals for quite a while, and he saw what you can see. And that is that these animals always swim forward. We never saw them swim backwards. And so he wondered whether or not this predominant anterior to posterior visual motion could serve as an instructive stimulus for topographic map formation. And he tested this in a very simple experiment. Well, simple in concept, not simple in execution. What he did was label single retinal ganglion cells within the eye of these animals, and then he put the animals in a chamber and provided them with an anterior to posterior moving stimulus or a posterior to anterior moving stimulus for 10 hours a day for 4 days. After which he assayed the resolution or the topographic map by mapping out the positions of the labeled retinal ganglion cells within the eye, here, along the nasal to temporal axis, and he mapped out the position of those axon arbors along the rostral caudal axis within the optic tectum. And what he found is shown here, where in animals, so on the y-axis is the axon projection site, and on the x-axis is the position of the retinal ganglion cell body. So what we know from this is that temporal retinal ganglion cells project to the rostral portion of the optic tectum, and nasal retinal ganglion cells project to the more caudal portion of the optic tectum. And when animals receive this anterior to posterior moving stimulus, they establish topography. But when animals receive the posterior to anterior moving stimulus, there is no topography within the projection of the retinal ganglion cell axons. And that means that if there was information sent from temporal retinal ganglion cells, it would terminate in the same part of the optic tectum as retinal ganglion cells from nasal retina communicating information. So there is no topographic information in this projection. And I should say, that this is the situation at the beginning of the experiment. So at the first time point, all of the animals had this level, i.e. no topographic information within their map, and the anterior to posterior stimulus allowed them to have this topography, and the posterior to anterior stimulus prevented it. So basically, the way that we think of this is that anterior to posterior motion stimulus enhances the development of the topography. And posterior to anterior degrades it or causes it not to develop. So we think of this basically as a simple example of temporal coding. There's information within the sensory periphery, which is then communicated in a temporal sequence to the retinal ganglion cells in a nasal to temporal, in a temporal to nasal pattern. And it's this activity within the retinal ganglion cells which then communicates the position, the relative position to terminate within the optic tectum. So what this meant was that if the retinal ganglion cells in the temporal retina fire first with this anterior to posterior motion stimulus, and that says, you terminate, those retinal ganglion cells terminate in the most rostral region of the optic tectum. And any ganglion cell that fires next would terminate sequentially more caudally within the target area. So we wanted to test whether or not this idea of the temporal sequence of ganglion cells determining where they're going to terminate within the optic tectum. And so the way that we tested this was by going back to the duly innervated optic tectum model that had been developed before. Because we know that if we ablate one optic tectum, as shown here on the right, that inputs from both eyes will terminate in the same tectum. And this would allow us to then stimulate the two eyes with very tight temporal intervals between the two eye stimulation and determine whether or not this affected the distribution of axons within the tectum. And again, the hypothesis is that whichever eye, the inputs from whichever eye is stimulated first will terminate more rostrally than the inputs from the eye which is stimulated second. And so we labeled retinal ganglion cells, put them into a little chamber where their eyes were stimulated at either 15 millisecond intervals, 50 millisecond intervals, or longer. And what we found was that when the retinal ganglion cells of the eye that was stimulated first, they would shift towards more rostral positions within the optic tectum, and the inputs that were stimulated second would shift to relatively more caudal positions. And so we call this a spatial to temporal to spatial transformation. And basically, the reason why we think this is important is because it would ensure that retinal ganglion cells' axons would terminate across the entire target area and not be squished up into the rostral portion of the optic tectum, as would be predicted from spike timing dependent plasticity rules. So here what you see is that the retinal ganglion cells would be activated in sequence, and according to this sequential activity pattern, they would terminate in a topographic manner. So just to compare this, what we're calling this spatial to temporal to spatial transformation, the STS, with the Hebbian coactivity rule. What Hebb said is all that really matters is whether or not you're coactive. And if the inputs are coactive, they will be strengthened onto the same target cells. So what we would like to add to that is that it really depends on the sequence of the input activity. And if they are coactive and offset by a certain temporal interval, then they will be adopt an order within the target area. And again, why is this important? It's because spike timing dependent plasticity would shift the topographic map towards one end of the target area, and the STS rule would ensure that this is spread out over time. So let's just look at this, and if we have activity going along this dimension. According to the spike timing dependent plasticity rule, the retinal ganglion cells that are activated first would capture all the territory, and they would have strong synapses with all of those targets. But the STS rule now would ensure that these connections are spread evenly from each input across the target area. I think this is interesting in light of recent work looking at spontaneous waves of activity in the retina, where we now know that these spontaneous waves originate in temporal retina and spread to nasal retina. And recent work in Michael Crer's lab has demonstrated that these spontaneous waves moving from temporal to nasal retina are present in the ganglion cell axons within the superior colliculus, and you can see that there's a wave of activity from rostral to caudal superior colliculus, both in the axons and in their postsynaptic cells. So I think that this is basically a conserved mechanism that organizes these topographic maps. So to summarize this part of the story, what we've shown is that the temporal order of convergent input activity is a source of information which organizes topographic maps, And the spatial information which is embedded in the natural visual scene in the form of optic flow is translated into a temporal code in the sequence of retinal ganglion cell firing, and that this temporal activity is then transformed into the spatial map of within the topographic map. And the reason I think that this SDS rule cooperates with spike timing dependent plasticity rules. Spike timing dependent plasticity rules are still very important in terms of allowing an animal to anticipate a response to a frequently observed input, but the SDS rule will counteract the negative consequences that spike timing dependent plasticity has on topography. So that story was all about axons and topography and completely ignored the other part of the story, which is basically that there are tectal neurons and that they're the ones who are processing this information. And so now I'd like to talk a little bit about experience-dependent properties of inhibitory neurons. And this was a project done by Hahn in my lab. So the postsynaptic cells, the tectal neurons, can be imaged beautifully in these neurons by using methods of single cell or sparse electroporation. And what you see here in this on the right are a series of 2-photon images through the depth of the optic tectum. What you see here now is the reconstruction of that same cell, and we can look closely at the dendritic arbor structure and how that changes over time during development and in response to visual input. When we collect time-lapse images in the intact animal, here at every 2 hours over a period of about 8 hours, it's interesting that the dendritic arbor doesn't really change a lot at this time scale, but over a time course of days, you see that the arbor elaborates. And we spent a lot of time investigating why there's so much dynamic in the short time frame without any net growth. Another way of looking at that is shown here, where in these optic tectal neurons, images collected every 10 minutes over a period of hours show that basically every process is dynamic. But remember, these animals are forming connections, they're processing visual information. So we wanted to understand how visual experience modifies the growth and plasticity of these neurons. And inspired again by Hüvel and Wiesel, we provided them with visual experience where we put them into a chamber where over the top of the chamber there was a panel of LEDs that were moving at a constant rate. And again, in a very simple experiment, we found that by imaging single neurons, and I'm showing you 2 examples here and here, by imaging single neurons over time, we found that when animals are placed, so what we do is we collect an image of the neuron in the intact animal, we take him off the stage of the scope, he swims around, and in the first 4-hour period, he's just sitting there in the dark, and then we collect another image. And you may see that the structure of the dendritic arbor doesn't really change a lot over this 4-hour period in the dark, but when the animal is now brought out of anesthesia and put into a chamber with that simulated motion stimulus, the dendritic arbor elaborates quite a bit. And so we did a number of experiments to identify many of the molecular and cellular processes that are involved in this visual stimulation-induced structural plasticity. Among others, we know that visual stimulation increases retinotectal synaptogenesis and increases the strength of these synaptic contacts by trafficking of AMPA receptors into those sites. So what we find, again, just to summarize these early studies, that immature neurons have very sparsely arborized dendritic arbors, and 2 things happen over the course of the development. The arbor gets more complex, and AMPA receptors are added to these synapses. And we were interested in dissecting whether or not AMPA receptors were, whether the arbor just grew and added and strengthened synapses, or if glutamatergic synaptic activity was actually important in the elaboration of the dendritic arbor. To address this question, we interfered with AMPA receptor trafficking into synapses and found that these neurons had a very different dendritic arbors, that they didn't develop mature synaptic contacts, and in fact they didn't respond to this simulated motion stimulus with an increase in dendritic arbor elaboration. So for instance, in the green bars, you see an increase in total dendritic branch length with visual stimulation compared to the dark. But in animals that had been, where the AMPA receptor trafficking had been blocked, these animals now, or these neurons, didn't show an enhancement in dendritic arbor growth in response to the visual stimulation. So in those experiments, we just went in blind and labeled and imaged cells independent of what kind of neuron they were. And so Hahn Hu was interested in testing whether or not excitatory and inhibitory neurons respond differently to visual experience. And the way that she did this was first by collecting time-lapse images of the neurons in the animal and determined whether or not they were excitatory or inhibitory neurons by using post-hoc immunocytochemistry for GABA. And part of the reason why we did this was that these neurons, they express GABA. We can identify them according to that immunoreactivity, but they don't express classical calcium binding proteins that inhibitory neurons express later on in development. So, and they also, according to studies that Carlos Eisenman has done, they don't have electrophysiological properties that would distinguish them from excitatory neurons. So when Hahn lined up all the inhibitory neurons and all the excitatory neurons that she had imaged, we couldn't identify any morphological difference between these two classes of cells. So by biomarkers, by biochemical markers, by morphology, by electrophysiological properties, these neurons, the excitatory and inhibitory neurons, appeared completely uniform at this relatively early stage of development. But we also knew, as I had told you, that this is a circuit that is seeing and processing visual information. So Hahn resorted to testing whether or not these neurons would behave differently in response to visual stimulation. So she put them through this now classical dark light visual stimulation protocol and examined whether or not how the inhibitory neurons responded. And what you see here are, this is again the change in total branch tip number and change in total dendritic length. And you look at these data, and it's like, oh my heavens. I mean, you see huge variation. And so what that meant to us right away was that this population of inhibitory neurons is extremely heterogeneous. And then what you see in this panel is when Hahn plotted the data so that she distinguished the growth rate of the dendritic arbor in response to visual stimulation and the change in growth rate that occurred when the animals were in the dark, then we see something very interesting that was quite distinct from the excitatory neurons that we had seen before. So neurons that increase their dendritic arbor growth rate with visual stimulation got smaller in the dark. And then there was another, and that's what classically the excitatory neurons do. But then we see this population of neurons where their dendritic arbor plasticity, structural plasticity, increased in the dark and decreased with visual stimulation. So for us, this was a total shock and suggested to us that there might be two classes of inhibitory neurons that had these different plasticity responses to visual experience. And so Hahn used this clustering strategy and was able to distinguish two groups of inhibitory neurons which behave in opposite in opposite ways in response to the dark and the visual stimulation. So this is the clustering, and again, this is that same plot that I just showed you, but now according to the clustering, these are separated into two different groups. And now what we see is they behave very nicely as two completely different groups with relatively little heterogeneity, where one group gets smaller in the dark and grows a lot with the visual experience, and the other enhances its structural plasticity in the dark and gets smaller with the visual stimulation. These two cell types are completely overlapping, so they're not segregated in any way within the optic tectum. So the reason why the dendritic arbor is basically a proxy for looking at synaptic inputs, because each little branch within the dendrite is basically like a spine in hippocampal neurons, where it's the site of a synaptic input. So when there's an increase in dendritic arbor branches, that's a proxy for an increase in synaptic contacts, and when there's fewer branches, it's a proxy for a decrease in synaptic contact. So now Hahn wanted to look at what about the output of the cell, and so we know that we can use calcium imaging because the magnitude of the calcium response correlates with the number of action potentials that the cell fires. So here what we see is, again, using the same dark light protocol, we find that neurons show differences in their calcium responses, and they fall into two groups. So again, the protocol here is provide a visual stimulus to the animal and collect the calcium response in response to that visual stimulus, put the animal into the dark, and then collect another calcium response and test whether or not it's altered as a result of the animal being in the dark, and then provide the animal with visual experience and collect another response. And what we find is, again, here, that neurons fall into two different groups, some of which potentiate their response to the visual stimulus as a result of the short-term visual experience that we show, and others depress their responses to the visual experience. And again, here it breaks down those two different groups. And so what these experiments indicate to us that there are two groups of inhibitory neurons, and they change both their input and their output in opposite ways in response to the short-term visual experience. Now, we were interested in what would happen to the balance of inhibition and excitation. What difference does this make for the circuit, basically, if you have these two classes of inhibitory neurons that might counterbalance one another? And so what we did was test what the effect of this short-term visual experience was on the E to I balance when we record from optic tectal neurons in the intact animal. And what we found was that animals that had been treated with the short-term visual experience for 4 hours, they had potentiated excitatory and inhibitory responses, as you see here in these examples, and here for the population of neurons from which we recorded. But importantly, the ratio of excitatory to inhibitory input was not different as a result of this short-term visual experience. So basically, the E to I balance in the optic tectum was preserved, and the plasticity kept this E to I balance. Okay, so how could that work? So basically, what we were trying to do a bunch of little stick figure cartoons of what kind of circuit might allow for two different classes, two opposite functional classes of inhibitory neurons and a single class of excitatory neurons that would allow for a balance of inhibition to excitation. And just to jump to our conclusion, what we propose is that excitatory input to the postsynaptic cell is balanced by a feed forward disinhibitory connectivity map or connection between the group 2 inhibitory neurons synapsing onto the group 1 inhibitory neurons. So basically, this little cartoon would accommodate these two classes of inhibitory neurons and that they would work together in order to maintain the excitatory to inhibitory balance within the optic tectum in response to visual experience. And so what this indicates to us is that we've identified at this early stage of development, prior to the time when there are morphological or biochemical clues to distinguish these two types of inhibitory neurons, they cluster according to their responses, their plasticity responses to visual input. And we think that these two classes of inhibitory neurons operate to maintain stability in the circuit at a time when there's a lot of dynamic rearrangement of neurons. We think that these two groups of inhibitory neurons are clear and functionally apparent prior to the expression of classical electrophysiological or biochemical markers that distinguish different classes of inhibitory neurons. If we look in these animals about a month later in development, all of these classical markers are present, and these neurons still serve this same purpose. So we think we have a window into this earlier stage of development than many people get to look at. And then the other interesting idea that we think is that these neurons acquire their different identities according to their position in the connectivity map within the nascent circuit. So again, I'd just like to thank the people in the lab. So this is Masaki, and this is Haiyan. This is Wanhua Shen, who did the electrophysiology in that last set of experiments. And again, thanks to the NIH and for funding, and to you for being here. Are there questions? If you have questions, I can answer them, either now or at the reception. I don't know, what's typical?
We can move to the reception.
Oh, okay. All right. Dr. Lee says, let's move to the reception.
Oh, thanks, Dr. Holly Klein for the wonderful lecture. So there's a reception in the library, and you can talk to Dr. Klein.